Today, we are jumping into a technology that honestly sounds like it's pulled from the pages of a sci-fi novel. It's called TimeVault, and it lets scientists read a living cell's past so they can understand and maybe even predict its future.
So to really get our heads around this, let's start with a problem that's been a frustrating, high-stakes puzzle for doctors and researchers for a very long time.
Picture this scenario. A patient gets a cutting-edge drug, one that's designed to attack their specific cancer. And at first, it seems like a miracle. But then, the treatment just stops working. The cancer comes roaring back. So why does that happen? What is it that lets this tiny group of cancer cells survive an all-out assault that kills nearly everything else?
Well, these survivors have a name: persister cells. And what's wild is that they're not resistant because of some new genetic mutation. Nope. Instead, they just hunker down. They enter this kind of deep survival mode, a temporary drug-tolerant state that lets them ride out the storm, ready to wake up and cause trouble later.
And we are not talking about a handful of cells here. No way. In studies on a common lung cancer therapy, we're looking at 20 to 25% of the cells switching into this persister state. That's a huge potential reservoir for a relapse.
So here's the real head-scratcher. To get the upper hand, you need to know what makes these cells tick. Are they just regular cells that get changed by the drug, or were they different all along? To really beat them, you'd need a way to spot their secrets before you even begin the therapy.
And that leads to the billion-dollar question. How on earth can you know what a cell was doing in the past before it ever saw a drug? It's like trying to figure out why a ship sank by only looking at the wreckage. You'd need a time machine.
Well, what if I told you that researchers at the Broad Institute of MIT and Harvard have basically built one? Let's take a look at this incredible new tool.
It's called TimeVault. In a nutshell, it's a synthetic gadget you can put inside a living cell. At a specific moment, it takes a snapshot of every single gene that's active, the cell's entire transcriptome, and then it stores that information right there inside the cell, safe and sound, so you can read it later. Honestly, the best way to think about it is as a molecular flight recorder. Just like the black box in an airplane, it captures all the crucial data about what was going on right before a major event, like a drug treatment. That way, later on, scientists can retrieve that recording and see exactly what led a cell to either survive or die.
All right, so how does this cellular time machine actually work? This is where the bioengineering gets really, really cool.
The amazing thing is, the team didn't have to invent this from scratch. They cleverly repurposed a structure that our own bodies already make. It's called a Volt particle, this tiny, hollow, barrel-shaped container. We're still not entirely sure what its day job is, but scientists realized it was the perfect little capsule for a recording device.
So here's the play-by-play. They start with a natural vault, and they engineer it, adding a special piece that's designed to grab onto all the cell's active messages. It's mRNA. Then, when they give the signal, these vaults just assemble on their own. And as they snap shut, they trap a perfect sample of all the mRNA inside the cell at that very moment. They become these tiny, durable time capsules, protecting that genetic snapshot for days.
And when I say durable, I really mean it. A normal, unprotected piece of RNA in a cell is incredibly fragile. It has a half-life of about 17 hours. But the RNA tucked away inside a time vault? Whoa! Its half-life jumps to over 132 hours. That's a massive, 7-fold increase in stability. The recording is built to last.
But, a recording is pretty useless if it's all garbled, right? Well, that is not a problem here. When the researchers checked, the snapshot stored in the time vaults matched the cell's actual state with over 90% accuracy. It's a crystal-clear, high-fidelity recording of the past.
Okay, so now they have this incredibly powerful new flight recorder. What do they do with it? They go right back to that original nagging mystery, the cancer persister cell.
They set up the key experiment. They took lung cancer cells, put the TimeVault system inside them, and then they hit record, capturing a snapshot of the cells while they were completely drug-naive, that is, before they had ever been exposed to the therapy. Only then did they introduce the cancer drug. After 4 days, just as expected, most of the cells were gone, but a small population of persisters survived. And this was the moment of truth. The scientists collected only those survivors, cracked open their time vaults, and read the recordings from the past. For the very first time, they could see what made these specific cells different from the very start.
And what they found was basically a smoking gun. A clear signature. The cells that were destined to survive were already running on a different metabolic engine.
Think of it like they were using a slow-burning backup generator instead of the main grid. They were also in a slower, more chilled-out state, just conserving energy.
And this just changes everything. It means the drug doesn't create persister cells, it just reveals them. They were there all along, this small group of cells with a unique survival strategy, hiding in plain sight.
And here's the proof in the pudding. This is where it gets incredibly powerful. Once they knew the persister's secret playbook, they could actually counter it. You can see here, when they combine the original drug with a second one designed to shut down that backup energy source, bam, they killed up to 62% more of the persisters. They literally used the cell's past to find its Achilles heel.
So let's zoom out for a second, because this is so much bigger than just this one cancer study. This is a fundamentally new way of looking at biology itself. I mean, listen to how the researchers themselves put it in their paper. They're not just talking about one small finding, they're talking about a powerful tool for studying the dynamic, moment-to-moment changes that drive all of life. It connects the cell's past to its future. Just let your mind run with the possibilities here. What could we ask now? How does a single stem cell actually decide to become a heart cell or a brain cell? What are the absolute first changes that lead to a disease like Alzheimer's way before symptoms show up? TimeVault gives us a real shot at finding those answers. It really leaves you with this one kind of mind-blowing thought. For so long, we've been trying to understand the story of life by looking at single frozen frames. But life isn't a photograph, it's a movie. And for the first time with TimeVaults, we finally have a way to hit rewind. Just imagine what else we're going to find.